The citric acid cycle, CAC, also known as the Krebs cycle or the TCA cycle, tricarboxylic acid cycle, is a series of chemical reactions to release stored energy through the oxidation of acetyl-CoA derived from carbohydrates, fats and proteins. The Krebs cycle is used by organisms that respire, as opposed to organisms that ferment, to generate energy, either by anaerobic respiration or aerobic respiration. In addition, the cycle provides precursors of certain amino acids, as well as the reducing agent, NADH, that are used in numerous other reactions. Its central importance to many biochemical pathways suggests that it was one of the earliest components of metabolism and may have originated abiogenically. The name of this metabolic pathway is derived from the citric acid, a tricarboxylic acid, often called citrate, as the ionized form predominates at biological pH, that is consumed and then regenerated by this sequence of reactions to complete the cycle. The cycle consumes acetate in the form of acetyl-CoA and water, reduces NAD+ to NADH, releasing carbon dioxide. The NADH generated by the citric acid cycle is fed into the oxidative phosphorylation electronic transport pathway. The net result of these two closely linked pathways is the oxidation of nutrients to produce usable chemical energy in the form of ATP. In eukaryotes, the citric acid cycle takes place in the matrix of the mitochondria, just like the conversion of pyruvate to acetyl. In prokaryotes, these steps both take place in the cytoplasm. The citric acid cycle is a closed loop. The last part of the pathway reforms the molecule used in the first step. The cycle includes 8 major steps. German chemist Hans Adolf Krebs' discovery of this cycle in 1937 marked a milestone in biochemistry. Krebs received the Nobel Prize for Physiology or Medicine in 1953 for this contribution to the study of intermediary metabolism in the oxidative breakdown of carbohydrates. Krebs and his co-author William Arthur Johnson published their findings, the role of citric acid in intermediate metabolism in animal tissues in enzymologia after being rejected by nature. That original publication was followed by many more. The TCA cycle is usually described beginning with acetyl-CoA, top position. Follow the diagram clockwise in the direction of the arrows. In the first step of the cycle, acetyl-CoA combines with a 4-carbon acceptor molecule, oxaloacetate, to form a 6-carbon molecule called citrate. After a quick rearrangement, this 6-carbon molecule releases 2 of its carbons as carbon dioxide molecules in a pair of similar reactions, producing a molecule of NADH each time. The enzymes that catalyze these reactions are key regulators of the citric acid cycle, speeding it up or slowing it down based on the cell's energy needs. The remaining 4 carbon molecules undergo a series of additional reactions, first making an ATP molecule, or in some cells a similar molecule called GTP, then reducing the electron carrier FAD to FADH2, and finally generating another NADH. This set of reactions regenerates the starting molecule, oxaloacetate, so the cycle can repeat. Overall, one turn of the citric acid cycle releases 2 carbon dioxide molecules and produces 3 NADH, 1 FADH2, and 1 ATP or GTP. The citric acid cycle goes around twice for each molecule of glucose that enters cellular respiration because there are 2 pyruvates, and thus 2 acetyl-CoAs made per glucose. These intermediaries are numbered on the diagram below. 1. Citrate. 2. Isocitrate. 3. Oxoglutarate. 4. Succinyl-CoA. 5. Succinate. 6. Fumarate. 7. Malate. 8. Oxaloacetate, oxaloacetic acid. Krebs cycle steps. You've already gotten a preview of the molecules produced during the citric acid cycle. But how exactly are those molecules made? We'll walk through the cycle step by step, seeing how NADH, FADH2, and ATP, GTP are produced and where carbon dioxide molecules are released. In the first step of the citric acid cycle, acetyl-CoA joins with a 4-carbon molecule, oxaloacetate, releasing the CoA group and forming a 6-carbon molecule called citrate. Step 2. In the second step, citrate is converted into its isomer, isocitrate. This is actually a 2-step process involving first the removal and then the addition of a water molecule, which is why the citric acid cycle is sometimes described as having 9 steps rather than the 8 listed here. Step 3. In the third step, isocitrate is oxidized and releases a molecule of carbon dioxide, leaving behind a 5-carbon molecule, ketoglutarate. During this step, NAD+ is reduced to form NADH. The enzyme catalyzing this step, isocitrate dehydrogenase, is important in regulating the speed of the citric acid cycle. The fourth step is similar to the third. In this case, it's ketoglutarate that's oxidized, reducing NAD+ to NADH and releasing a molecule of carbon dioxide in the process. The remaining 4-carbon molecule picks up coenzyme A, forming the unstable compound succinyl-CoA. The enzyme catalyzing this step, ketoglutarate dehydrogenase, is also important in regulation of the citric acid cycle. In step 5, the CoA of succinyl-CoA is replaced by a phosphate group, which is then transferred to ADP to make ATP. In some cells, GDP, guanosine diphosphate, is used instead of ADP, forming GTP, guanosine triphosphate, as a product. The 4-carbon molecule produced in this step is called succinate. In step 6, succinate is oxidized, forming another 4-carbon molecule called fumarate. In this reaction, 2 hydrogen atoms with their electrons are transferred to FAD, producing FADH2. The enzyme that carries out this step is embedded in the inner membrane of the mitochondrion, so FADH2 can transfer its electrons directly into the electron transport chain. In step 7, water is added to the 4-carbon molecule fumarate, converting it into another 4-carbon molecule called malate. In the last step of the citric acid cycle, oxaloacetate, the starting 4-carbon compound, is regenerated by oxidation of malate. Another molecule of NAD+ is reduced to NADH in the process. Here is a view of the steps mentioned previously. Key TCA cycle enzymes: malic dehydrogenase, ketoglutarate dehydrogenase, citrate synthase, fumarase, aconitase. One TCA cycle turn yields 7 products: GTP, 3-NADH, FADH2, which is converted to UQH2 in the presence of coenzyme Q, ubiquinone, 2-CO2, carbon dioxide. Because 2 acetyl-CoA molecules are produced from each glucose molecule, 2 cycles are required per glucose molecule. Therefore, at the end of 2 cycles, the products are 2 GTP, 6 NADH, 2 FADH2, and 4 CO2. The above reactions are balanced if Pi represents the H2PO4- ion, ADP and GDP, the ADP2- and the GDP2- ions respectively, and ATP and GTP, the ATP3- and GTP3- ions respectively. The total number of ATP molecules obtained after complete oxidation of one glucose in glycolysis, citric acid cycle, and oxidative phosphorylation is estimated to be between 30 and 38. Pyruvate is a biological molecule and product of glucose metabolism that reacts with adenosine triphosphate and carbon dioxide, converting it into acetyl-CoA and adenosine diphosphate (ADP) at the beginning of the TCA cycle. It is often included in the introductory or preliminary step of the cycle. Pyruvate usually derives from the glycolysis product pyruvic acid, which quickly dissociates in most natural systems, leaving pyruvate. Pyruvate plays an important role in multiple biotransformations, as explained in greater depth in the article Pyruvate Transformations. Allosteric regulation by metabolites. The regulation of the citric acid cycle is largely determined by product inhibition and substrate availability. If the cycle were permitted to run unchecked, large amounts of metabolic energy could be wasted in overproduction of reduced coenzymes such as NADH and ATP. The major eventual substrate of the cycle is ADP, which gets converted to ATP. A reduced amount of ADP causes accumulation of precursor NADH, which in turn can inhibit a number of enzymes. NADH, a product of all dehydrogenases, is the citric acid cycle with the exception of succinate dehydrogenase, inhibits pyruvate dehydrogenase, isocitrate dehydrogenase, ketoglutarate dehydrogenase, and also citrate synthase. Acetyl-CoA inhibits pyruvate dehydrogenase, while succinyl-CoA inhibits alpha-ketoglutarate dehydrogenase and citrate synthase. When tested in vitro with TCA enzymes, ATP inhibits citrate synthase and ketoglutarate dehydrogenase. However, ATP levels do not change more than 10% in vivo between rest and vigorous exercise. There is no known allosteric mechanism that can account for large changes in reaction rates from an allosteric effector whose concentration changes less than 10%. Citrate is used for feedback inhibition, as it inhibits phosphofructokinase, an enzyme involved in glycolysis that catalyzes formation of fructose 1,6-bisphosphate, a precursor of pyruvate. This prevents a constant high rate of flux when there is an accumulation of citrate and a decrease in substrate for the enzyme. Regulation by calcium. Calcium is also used as a regulator in the citric acid cycle. Calcium levels in the mitochondrial matrix can reach up to the tens of micromolar levels during cellular activation. It activates pyruvate dehydrogenase phosphatase, which in turn activates the pyruvate dehydrogenase complex. Calcium also activates isocitrate dehydrogenase and ketoglutarate dehydrogenase. This increases the reaction rate of many of the steps in the cycle and therefore increases flux throughout the pathway. Transcriptional regulation. Recent work has demonstrated an important link between intermediates of the citric acid cycle and the regulation of hypoxia-inducible factors, HIF. HIF plays a role in the regulation of oxygen homeostasis and is a transcription factor that targets angiogenesis, vascular remodeling, glucose utilization, ion transport, and apoptosis. HIF is synthesized constitutively, and hydroxylation of at least one of two critical proline residues mediates their interaction with the von Hippel-Lindau E3 ubiquitin ligase complex, which targets them for rapid degeneration. This reaction is catalyzed by prolyl 4-hydroxylases. Fumarate and succinate have been identified as potent inhibitors of prolyl hydroxylases, thus leading to the stabilization of HIF. The main function of the Krebs cycle is to produce energy, stored and transported as ATP or GTP. The cycle is also central to other biosynthetic reactions where the intermediates produced are required to make other molecules, such as amino acids, nucleotide bases, and cholesterol. The Krebs cycle is found in all cells that use oxygen. Combined with the process of oxidative phosphorylation, the Krebs cycle produces the majority of energy used by aerobic cells, with the percentage energy provided for humans being greater than 95%. The TCA cycle was first observed in the muscle tissue of a pigeon. It takes place in all eukaryotic and prokaryotic cells. In eukaryotes, it occurs in the matrix of the mitochondrion. In prokaryotes, it takes place in the cytosol. Thank you for watching and have a great day.